Right. So yeah, thank you for the invitation. I want to say I was part of the very initial sort of stages of getting the BioNet together, and I'm glad to see it has picked up a momentum, and there's all these events happening on a regular basis now. And yeah, thank you everybody for being here. Today I will talk to you about my work on the evolution of sexual asymmetry, which is work that I started during my PhD years at UCL. And this has been a field that I have remained active since, even though I became interested in modeling developmental biology as well. So, and I don't get to talk too much about this work, in fact, so I'm very excited about the opportunity to do so today. Right. Oop. Up. Yes, that's it. So the work I will present today is very much motivated by the observation that in the striking majority of sexual organisms, the two gametes or sexual cells that fuse together to make a zygote are not the same as one another. So this asymmetry is more obvious in oogamous species where females produce a large gamete, the egg, and males produce the small sperm, as you can see in the image here. But it's also true in isogamous species like yeasts and ciliates and chlamydomonas, where the two gametes fusing together are morphologically the same, but they differ genetically, and only individuals that belong to different sex groups can reproduce together. So in these cases, we don't really talk about sexes anymore, but of isogamous mating types. An example is the ciliate tetrahymena. You can see here the picture of the two gametes fusing together that appear to be exactly the same, but they differ genetically and in their sort of mating type identity. So this asymmetry seems to be fundamental to sexual reproduction. And a lot of the work I will present today asks why is this the case? And what are the evolutionary origins of sexual differentiation? And also, what are the implications of this requirement for asymmetry in mating for other features we see in the sexuality, particularly in unicellular organisms? And understanding the evolution of this asymmetry in isogamous species that have mating types like chlamydomonas that you see here in the bottom left picture is fundamental because it's thought that's where the origins of sexual asymmetry as we see it in oogamous species can be traced. The idea is that once mating types have evolved, the basis to evolve anisogamy, oogamy, and morphological asymmetry is established. So to address the question of the evolution of mating types, I began to ask myself, what is it that mating types do? What is their function? And how can they do that best? And this led to a lot of reading to try and figure out what are the sort of universal, if they exist, functions of mating types that are shared between unicellular organisms across the tree of life. And I want to take a few moments here to go through these general features of the life cycles of unicellular organisms and the key steps that are required for sexual reproduction. Now, most of the time, these organisms actually grow asexually through mitotic division, and at some point in their life cycle, probably triggered by stress, they arrest growth and they differentiate into sexually competent cells or gametes. This is often an irreversible process. So to re-enter this mitotic growth phase, cells actually need to find a partner and fuse and undergo meiosis. And to do this, cells need to find a partner. And this often requires signals that are diffusible that allow gametes to locate each other and move towards each other. Now, once cells meet each other, they stick together and they undergo fusion. And finally, cells undergo meiosis to give rise to the new generation that can go on again and grow asexually. Now, for all of these steps to occur robustly and efficiently, there's a lot of communication between cells that takes place. Sorry? Okay, no, I thought it was a question. So now for all of these steps to take place robustly and efficiently, there's a lot of communication that has to happen between cells, both between the two cells that are actually mating and at the population level. For example, the entry into gametogenesis often requires this phase of arresting growth and becoming gametes, sexually competent cells, requires cells to sense the presence of conspecifics in the environment, to know that there are potential partners to mate with once this differentiation step takes place. And this can happen when cells secrete pheromones to indicate their presence. And likewise, for partner finding, cells use chemotaxis to indicate their location and to find partners. And the process of cell fusion also requires signaling interactions that allow cells to remodel their cell walls and fuse. So in all of these steps, the signaling interactions between pairs of cells happen in a mating type specific manner, so that the ligands or signals produced by one cell type induce a response only in cells of different mating type. And the hypothesis driving my work is that these interactions between cells are actually optimized when they operate asymmetrically. And we can trace the evolution of sexual asymmetry on this selective pressure. And to test this hypothesis, I began to ask how would signaling interactions between gametes be optimized? And I have been using theoretical models that help us think how the physical constraints that underlie cell-to-cell communication may affect the efficiency of mate finding and interactions between cells during sex. So as I mentioned now in a couple of slides ago, gametes often rely on sexual chemotaxis to find each other. So you can imagine that if there are no mating types and all cells send and produce the same chemoattractant or pheromone to find one another, finding a partner could be a lot of work. This is because as cells move, they would generate a field of pheromone that is their own pheromone that is the same as the pheromone that they would need to sense. And that would make it difficult for them to distinguish their own secreted signal from that of a potential partner. And to be more quantitative, one can write down the equations that describe this field, a concentration field of chemoattractant in space when several cells secrete it while they move. So here, this is the equation that captures this dynamics. This term here captures diffusion, degradation of the chemoattractant, and this is a localized source at the position of each cell at time t. So this would give you the concentration profile that would be available to cells to encourage their movement if they move chemotactically. And then we can assume that cells, or at least in this context, I'm making the assumption that cells sense the difference in concentration at their two edges to move. But I think similar arguments can be made for other chemotactic motions that are temporal rather than spatial. So what you quickly see is that as cells move, they induce a concentration, or a tail of high concentration behind them, as you kind of see in the cartoon here. And yeah, so yeah, if you simulate this system, you can and follow the tracks of pairs of cells, you can see that cells that send in response to the same pheromone, as you see in the figure here, are indeed much slower than finding a partner if you compare them to simulations where cells are divided into signal senders and signal receivers, if you like. And I would say that this result is somewhat intuitive, but this study also allowed us to think more quantitatively about the potential constraints to cell pairing in the absence of any asymmetry in sexual chemotaxis. And we were able to place these constraints in the context of physical parameters like the density of cells, the diffusivity of the pheromone, the rate at which it degrades, and so on. So this is thinking about these constraints of cell signaling in the context of chemotaxis. Now, mating cells need to also interact not just to find one another, but also to coordinate gametogenesis and cell fusion. And these processes don't necessarily require cells to respond to spatial cues. But what we know is that cells need to read one another's signals in a quantitative manner. For example, we know from Sophie Martin's lab at the University of Lausanne that feedback and amplification of signals are important for cell fusion in yeast during sex. So the question then becomes, what sort of constraints or costs do we expect to see in signaling interactions other than chemotaxis between mating cells when there is no asymmetry in this communication? And could we use our predictions about these potential costs to say whether they are sufficient to predict the evolution of self-incompatible mating types? And this is what this sort of next chapter of the talk will be about. So to do this, again, we wrote down equations that capture the dynamics of the free and bound ligand and receptor concentration within cells. So we're following again, ligands and receptors within cells. We're just not that we are assuming that these are essentially not diffusible anymore. So the way to capture these dynamics, I'm going to go through these equations to give you an intuition of what they mean. So the dynamics of the free ligand depend on the production rate, the rate at which the ligand binds the receptor within the same cell, the rate at which the bound compound becomes unbound, and the degradation rate. A similar equation holds for the amount of free receptor, and we can also follow the bound ligand receptor complex that increases at the rate at which the ligand and receptor bind. It's removed at the K-, which is the rate at which the bound complex unbinds, and it also degrades at the given degradation rate. So these equations we can solve at steady state to obtain expressions for the concentration of the free ligand, receptor, and bound complex. And we can plot this concentration at steady state as a function of this variable K plus, the rate at which the receptor and ligand would bind to one another within the same cell. And what you notice here is that there is a steep decline as the value of K plus, the rate at which ligands and receptors within the same cell interact. So there's a steep decline as this K plus increases, and it becomes steeper when the degradation rate is smaller. So these features, I think, are interesting to come back to when we discuss the evolution of the system and how free the design of these molecular interactions really is in practice. For now, I want to move on to describe how we studied the evolution of the production rates of different ligands and receptors under selection for quick and robust mating. First, we will assume that the strength and the precision of intracellular signaling is what determines mating success. And the strength of the signaling interaction between cell 1 and cell 2 will depend on how well cell 1 can read the signal that cell 2 sends, and vice versa. And we use this phenomenological description shown here that assumes that how well cell 1 reads cell 2 increases with the amount of signal cell 1 can receive from cell 2 and decreases with the amount of signal that cell 1 senses from itself. So this second term here is a cost, is a term that reflects a cost that is meant to be there due to interference in feedback and robustness due to self-bindings, due to the ligand and the receptor binding and potentially signaling within the same cell. So then the probability that two cells mate, right. So these animations were meant to come with that. The interference due to cost, and this is the strength of the ligand receptor interactions between cells. And another probability that two cells mate depends on the product of these two strengths of how well cell 1 reads cell 2 and how well cell 2 reads cell 1. And I'm using a function that this has this sort of form to reach a saturation point, essentially. Now, within this framework, we developed a population model where each cell could produce either two ligands or two receptors. As you see here in this little cartoon that I have the two ligands marked as capital L and little l, and the receptors marked as capital R and little r. And the assumption here is that only the capital R ligand would interact with the receptor that is represented by capital R, and the same for the lowercase letters. So the assumption here is these production rates undergo mutation, and we impose selection by only allowing cells that interact strongly with each other to mate, using the equations I've shown you in the previous slide. This gives us a framework to ask how we could move from a system that all the cells are the same, as shown in this panel here, to a system where the ligand and the receptor are produced in an asymmetric manner, but that divides the population into two types of cells that are unable to interact, two sort of groups of cells that are unable to interact within the same the group, but send their signals asymmetrically. And so the question is, how do we move from one configuration to the other? And to capture how the degree of symmetry in the system evolves, we define at the population level, we define this variable S, which is equal to 1 if all cells are uniformly producing the same ligand and receptor, as shown in panel A, and it becomes 0 if cells produce the ligand and receptor asymmetrically, as shown in panel 2. So this is a sort of measure that we can follow over time to understand how our population is moving towards or away from asymmetric mating, from asymmetric signaling and mating types. So what what we ended up seeing when we studied this system in an evolving sense is that there were two possible equilibrium states, what we call E1, where S is 1 and there is no, this is a fully symmetric system, no asymmetry in mating, and all the cells are the same with one another and can interact with one another equally. And we also saw a second equilibrium where S converges to 0, and as you see here, it's due to mutation and the finite population size. So in this second state is a fully polarized or asymmetric signaling state that is equivalent to having two mating types. And the question here is what determines which one of these two equilibria is stable. And I think it's also interesting here that we do not see an intermediate state where both of these, all three of these types are present. Right, so the question now is, what determines whether we're in equilibrium 1, where signaling is symmetric and we essentially don't have any mating types, versus equilibrium 2, where signaling is asymmetric and we have a population with two mating types. So sexual asymmetry has begun to evolve. So which one of these two states becomes evolutionarily stable actually depended in key parameters in our system that reflect physiological parameters in a putative biological system. So in particular, we saw a jump in which state was stable from one to the other based on two key parameters: gamma, which is the rate at which the ligands and the receptors degrade in our system, and K+, which is the rate at which ligands and receptors within a cell bind. And the asymmetric signaling of or two mating type equilibrium E2 is more likely to take place when K+ is higher. So when cells, when ligands and receptors within the same cell are more interact with each other more, and for larger values of gamma. Now the first thought here is that cells could just resolve all of these issues about signaling interference by simply setting K+ to 0. However, I would argue that it's not obvious how this would affect the strength of interaction between ligands and receptors, amongst cells. For example, in fungi and ciliates, cells secrete pheromones locally once they have found a partner to induce adhesion and fusion. And since in this case the ligand is not surface bound, reducing its affinity within a cell will also reduce its affinity between cells, which is bad for cell communication. And I want to also emphasize that this is a crucial part in the lives of these unicellular organisms, because any mistake that happens at the moment where cells are trying to fuse with one another and lyse their walls can lead to cell death, essentially. So I think it's not entirely trivial for cells to really abolish binding within the same cell without having an impact on between cell communication. And I would be interested to hear more what the cell biologists in the background think about this. Now, going back to our model, what I haven't sent to you so far is that I have made this assumption in the underlying genetics that the ligand and receptor loci are tightly linked. And although this is the case in mating types in nature, there is no reason to make this assumption a priori. So in this next step, I wanted to test the effect of recombination would have in our system. So if the receptor and ligand loci were able to recombine at the rate, let's say rho, how do our results and conclusions change, and what can we say about recombination as well and the evolution of recombination? So what we see is, again, two equilibrium states become possible when recombination is considered, but the asymmetric signaling state, where S was 0, now moves away from 0, meaning that cells that are sort of asymmetric, but not completely so in this case. And you can imagine that if recombination was present and you had full asymmetry, you're running the risk to end up with a cell that has no ligand or no receptor. And that's essentially the reason why cells prefer to move away from that state and be a little bit asymmetric, but not completely. And in fact, when the recombination rate is very high, the population converges to a state where cells are still divided into two types, in the sense that we see two possible configurations. As you see here, these are cells that produce both ligands but only one receptor, and cells that produce and cells that produce one receptor and sorry, both ligands and one receptor, and then there is a division between which one of the two receptors would be produced. Right. And this is a solution that evolves due to deleterious effects of recombination in our system. So you evolve some degree of asymmetry, but not quite what you really see in mating types. So, well, the obvious next step was to ask what happens when the recombination rate is also allowed to evolve together with the ligand and receptor production rates. And lo and behold, what we observe is that the coevolution of the recombination rate together with the signaling production rates. And what we see is that here we allow the recombination rate to fluctuate stochastically to begin with, while the signaling rate is not allowed to evolve at all, just to reach some sort of equilibrium in the recombination rate. And what we see is that once selection is imposed, the recombination rate begins to decline, and this follows a very sharp decrease in the signaling symmetry, so that we end up with tightly linked receptor and ligand loci with the ligand and receptor of the ligand receptor alleles encoded asymmetrically. And this was very exciting to see, to me, because the selection forces that reduce the recombination rate between ligand and receptor loci in mating types are thought to be behind the evolution of mating type loci, which are themselves possible precursors of sex chromosomes. So this sort of dynamics could perhaps help us understand of what was the very origins that planted the seed for the evolution of sex chromosomes in multicellular organisms. So what I have told you so far is that gametes can communicate with each other more efficiently when signaling interactions are polarized or asymmetric, and that the constraints that underlie cell-to-cell signaling may help explain the evolution of self-incompatible mating types. Recombination interferes with this asymmetry, but tight linkage between the ligand and receptor co-evolves together with asymmetric signaling roles in our system. Now, I have so far spoken to you about the evolution of mating types. Why would a population that has no mating types evolve mating types in the first place? And my focus has been so far a transition between no mating types to two mating types. However, there was no reason in theory to just consider only two mating types. And in fact, in many isogamous species that are sort of the inspiration or motivation of this work, have not just two, but several mating types. Although two mating types are common in algae and diatoms, the number of mating types can go from 2 up to 10 in ciliates, and some fungi can have thousands of mating types. So what determines the number of mating types, and how can the ideas that I have introduced so far can help us also explain the conditions under which several mating types evolve? So to address this, what we did was to think of signaling as a preference. So we think of signaling interactions between cells as a preference, so that a strong signaling affinity between two particular cell types is like a preference for a particular cell type. And this preference can presumably vary, can be under can mutate and be under selection. And the question in this sort of project is, how do the frequencies of different mating types in the population vary as a function of these preferences? So the assumptions we've made here is that cells have finite resources to put in signaling, so the total sum of preference or investment in a particular signaling interaction they can afford is finite. Then one can write down just classical population genetics equations that keep track of the frequency of each cell type. Here I'm showing you an example where you have 3 types that have the preference of cell 1 for cell 2 and preference of cell 2 for cell 1 and so on. And the probability that these two cells mate are a product of their frequencies times the a normalized product of the frequencies times the preference they have for one another. And we can use these equations here to study how mating type frequencies evolve. So here Fi is the frequency of mating type I. So this is a general system that you can use for I mating types, I being 2 or 10 or 100. And in this system here, I'm showing a simplified case where I is equal, takes values in 1, 2, and 3. And we ask how do these frequencies evolve. And we can solve these equations in the simple case where 3 mating types are possible. And we have 2 types of solutions. One where 2 mating types are stable. So either type 1 and 2, 1 and 3, or 2 or 3. Or a situation where we have 3 mating types being stable. And we can also solve these equations numerically and see all these different cases where we start with type 2 and 3, and 1 and 2, and mating type 3 is introduced and takes over type 1, or under different conditions, mating type 3 just reaches an equilibrium where 1, 2, and 3 are at the same frequency, or mating type 3 invades again, but in a sort of the frequencies of the 3 mating types are not symmetric. So the question now is, what determines when a population with 2 mating types evolves a 3rd mating type? What are the conditions that allow the invasion of a 3rd mating type? So allow this increase in the number of mating types. And it turns out that this depends on how strong the preference or signaling interactions between the 2 resident types are relative to the mutant 3rd type. This is what this equation essentially is telling you. So I want to now simplify this a bit further to try and give you a bit more intuition. Here I have assumed, for simplification purposes, that the preference of cell 1 for cell 2 and cell 2 for cell 1 is quantified by a number gamma. A 3rd type appears, so this is the mutant type that has a preference for both resident types 1 and 2 that is given by gamma plus eta for some quantity eta. And the preference of mating type 1 and 2 for this mutant are given by gamma plus kappa 1 and gamma plus kappa 2. So this phase diagram here shows you the space where the 3rd type invades. So the first insight here is that the value of eta does not matter. Mating type 3 can like the 2 resident mating types as much as it wants, but it will not invade unless it's also liked sufficiently by the residents. And in fact, if kappa 1 and kappa 2 are sufficiently small, the 3rd type will not invade. And what this means is that this new mutant, presumably carrying some alternation in its pheromone and receptor configuration, must have acquired mutations that did not impair its interactions with the resident types for it to spread. And although this is certainly possible, it does provide somewhat strict constraints for a 3rd type to evolve, and could perhaps explain why we don't always see an explosion in the number of mating types as one would expect. And of course, this argument can be made a bit inductively. So here I spoke to you about how you go from 3 mating types to 2 mating types. You can ask the same question and use a similar logic to understand how would you go from 3 mating types to 4 mating types, and a similar sort of logic follows in that case. I want to also mention that here that there is some really nice work from Hannah Coco's group in the University of Zurich, where the role of the population size itself and drift is shown to also explain how the number of mating types is constrained or evolves. And I think there's some interesting sort of complementary ideas with what I just told you here. Now, I have shared some ideas with you about why mating types may have evolved and factors that could determine their number. But cells need to also find a compatible partner, and this can cause problems. For example, in small or sparse populations or populations that disperse a lot, the mating type ratio can become distorted during asexual growth, which can actually, these organisms undergo asexual growth for long periods, most of their lives, in fact. And these fluctuations that can happen during these periods may mean that cells can find themselves without potential partners. And this can be particularly so in a population that has only 2 mating types, where, again, dispersal or random death of different cells can leave the population in a situation where, when cells are ready to mate, the population does not reside in a state where you have 50% of one mating type and 50% of the other type. One potential solution to this problem would be for individuals to switch their mating type and identity during mitotic division. And in fact, there is direct and indirect evidence in lots of different unicellular species, from yeasts to ciliates and chlamydomonas, that indeed cells can change or switch their mating type identity stochastically. And to understand this process, we studied the evolution of mating type switching in finite populations. To do this, first, we asked how the mating type ratio changes due to stochasticity during vegetative growth. So this is what you see in this panel here. You can see that the mating type ratio, which would ideally be 1, becomes increasingly distorted the longer the vegetative or asexual growth period is, and the smaller the population is. So this makes sense. Small populations are undergo more drift, and longer periods of this vegetative growth provides more stochasticity for this ratio to be distorted. We then allowed the capacity to switch mating type identity during asexual growth to be an evolvable trait quantitatively. And not very surprisingly, mating type switching evolved as a response to this pressure to find partners. And this is what you see in this panel here, where for different population sizes, you can see the fixation probability of a mutant that conveys switching for life cycles, for populations that have life cycles of varying vegetative growth periods. What was a bit more surprising in our analysis was that we observed a relatively low switching rate to be adaptable rather than high switching rates. And also, somewhat surprisingly, populations that had longer periods of asexual growth evolved lower switching rates. So here you see the switching rate itself and the fixation probability for populations that have different durations of asexual growth. So you can see here that as the asexual as the period of asexual growth increases, the fixation probability doesn't increase massively. And actually, in these populations with long periods of asexual growth, the switching rate just reaches an optimal 0.21 and doesn't increase much with further rounds of vegetative growth. And this was not immediately clear to us. And this is how we rationalize this observation. First, we see that the sex ratio declines sharply without any switching, but is maintained at very high levels, even for very small switching rates. So here you see the switching, sorry, the mating type ratio as vegetative for a given number of vegetative growth rounds for 0 switching rate. For 0 switching rate, and then for small incremental steps of increase in the switching rate. And you can see already a large improvement in the mating type ratio for a switching rate as small as 5%. And this additional improvement that happens as we incrementally increase the switching rate becomes infinitesimal. As you can see, the red line becomes closer and closer to the blue line, and the orange line and brown lines are very close to each other. So, and then in addition to this, in addition to the fact that higher switching rates don't convey a higher advantage for the mating type ratio, during asexual growth, there's actually no selection to maintain the sex ratio near 1. And so switching is in fact a neutral trade during that time. And someone could also argue that this is also a costly trade during asexual growth. And these considerations together can help explain why populations with life cycles that include longer periods of asexual growth, end up evolving lower switching rates. And this prediction that we make that a very low switching rate could in fact be sufficient to maintain a population at the optimal sex ratio or mating type ratio also suggests that it might not be completely trivial to detect experimentally the switching rates in populations like in species like yeast, that dispersal is very high, and we expect the mating type ratio to be distorted massively. The switching rate is expected to be higher, and it has indeed been observed to be 25% in those cases. But I would argue that in other species, that are consistently in larger populations, perhaps we need to look in more detail to see this experimentally. So in summary, stochasticity in small or sparse populations can distort the mating type ratio during vegetative growth, and low levels of mating type switching can be enough to mitigate this issue. This suggests that mating type switching may be more frequent than we thought, as it has only been observed in a handful of species explicitly or directly. For example, people have seen mating between cells of colonies that started from a single cell in some species of Chlamydomonas. And the conclusion here was that these species had no mating types. But I would argue that this could also be an indication of mating type switching that is yet to be directly observed in those populations. So with this, I want to summarize what I told you today. I told you that asymmetric signaling roles and mating types can evolve under selection for robust and efficient mating. And looking into the constraints at the cell biological level can be key to help us understand of where these selection pressures are coming from. In the system, linkage between the ligand and receptor loci evolves, and this could perhaps help us understand how sex chromosomes themselves have evolved, or the road to the evolution of sex chromosomes. And the number of mating types can also depend on this efficiency of cell-to-cell signaling. And longer periods of asexual growth can distort the mating type ratio, and this puts pressure on individuals to evolve the capacity to switch their mating type identity stochastically. And with that, I want to thank my collaborators in the projects I presented today. This was work I started as a PhD student. I did my PhD with Andrew Pionkowski and Nick Lane, and a lot of the work I continue doing with POM. The work on mating type switching was done with Bram Kushper, who was a postdoc with me at UCL, and the chemotactic work I presented at the very beginning, I did in collaboration with Yo Yuasa in Japan. And these are the institutes that have hosted me over the time and have funded me. And yeah, thank you for listening. I'm looking forward to taking questions.